Well, good evening, ladies and gentlemen. This is the Society for the Application of Research, and we could have not done better. In fact, our distinguished programme secretary, Andrew Shepherd, could not have done better than to bring us Billy Boyle this evening. Billy really epitomises the whole story of doing some research, being a student here, doing some research, turning it into a really successful business. So I think he needs very little introduction, and I'm going to let him introduce himself by what he does. And afterwards, we can cross-examine him. And afterwards, we can go to the bar. So you're all very welcome. Please welcome Billy Boyle.
Good evening, everyone. It's a great pleasure to be here. Thank you for the very kind introduction. I, too, am also enthusiastic about the bar afterwards. So what I'd like to do is tell you a little bit about our company, Owlstone Medical, how we got there, what we're doing, what the mission of the business is. And I really do want to give enough time and opportunity at the end to ask questions about anything that we've covered today. So our business is developing a breathalyzer for disease, and our company mission is to try and save 100,000 lives. And today we see two big problems in healthcare. The first is too many patients are diagnosed with disease when it's too late to cure, and too many people are on drugs that don't work for them. So these two big areas of early detection and precision medicine are where we're focusing our technology. So has anyone ever come across the headline, my dog has sniffed cancer, or dogs sniff cancer? It kind of pops up, on BBC website about once every 3 months. And what's behind that is this notion that there's chemical markers from the body, which are markers for disease and illness. So every time I breathe out, or anyone here breathes out, you have thousands of chemicals on your breath called volatile organic compounds, or VOCs. And some of these VOCs are specific markers in everything from cancer to infectious disease and inflammatory disease. So if you're able to build a breathalyzer device that can tease out these tiny chemical signals, you have a device that could make a big difference for patients in terms of early detection. So the technology that sits at the heart of what we do is a programmable microchip chemical sensor. So it's a bit like an electronic nose on a chip. We actually developed it back in Cambridge University in 2004. So me and two other researchers were in the dingy engineering basement. We were in the microchip division, and we had a big interest in the kind of convergence of new manufacturing techniques and sensor technologies where if you could reduce size, you could enable whole new classes of applications. And along that lines, there was a big need after 9/11 for better sensors for detection of explosives and toxic gases. And the three of us got together, came up with this technology idea. And around that time, there was tremendous amounts of support for students trying to spin businesses out from the university. We went through the business plan competition. The first ever investor that we pitched was actually in the audience back there, David. And I think we give the worst pitch that we ever did to David. But after that, we were able to raise financing, which spun us out of the university, out over into St. John's Innovation Centre, where we're trying to develop this chemical sensor technology for the detection of toxic gases and explosive threats. And over the years, we've been reasonably successful at that. So we reincorporated the business in the US, raised a lot of money primarily from US-based investors, but also won a lot of contracts from the likes of the US Department of Defense and Department of Homeland Security. And what this allowed us to do was develop and mature this technology, and we built that into a profitable business. And what makes the sensor special, it's not only small and low cost, but you can reprogram what you want to sniff just by changing the software on it. So you always have the same underlying technology. If you want to detect a different chemical, you just change the software. So we always had in mind, can we use this in diagnostics applications? We know we have these chemicals coming from breath and present in urine. Can we use technologies like this for these diagnostic applications? Now, the medical business is a completely different animal from the military business. So the investment requirement, what the exit's going to be. So we decided last year to split the medical business out as a completely separate entity. So since then, we've raised $23.5 million of investment, most recently from Aviva, which is great because they're a big healthcare insurer. They see the benefit that early detection can bring for patients. And I think, what we've been really beneficial of is being able to leverage this, well-proven, mature technology. It took a long time to develop and start to apply it in these types of applications. So today we've got 2 facilities here in Cambridge. So just over in the Science Park, we do all our own manufacturing, and we have a breath biopsy lab out there, which I'll talk about a little bit. So when I think about these breath chemicals, there's kind of 2 key questions in my mind. What are they and where do they come from? In terms of what are they, so if you look in your body, your genome tells you about the risk of getting disease. Your genetic code is telling you about disposition and risk. But it's actually your downstream metabolism that gives you the best real-time snapshot of your actual disease state. So what we're looking at, these chemical markers on the breath are metabolites. And it's really about that immediate snapshot of underlying state. And when you start to think about cancer, for instance, in early detection in cancer, we've known for close to 75 years that cancer has an altered metabolism even at the earliest stages. So if you're on the hunt for biomarkers for early stage disease, this is a good place to look. And then if you're thinking about respiratory type disease, so things like asthma and COPD, and you're trying to find out what are the best drugs that will work for my particular disease, these markers are good because they tend to relate to specific disease activity and not just the absence or presence of cells. So the second question is, where do they come from? Now, obviously a lot of them come from the airways themselves, so being produced locally within the lungs. But actually, when you think about what your lungs do, they're an extremely good mechanism of exchanging chemicals from the blood to the airways, CO2 and oxygen. So a lot of the chemicals which are coming out actually originate from your blood. And this is interesting because it opens up this possibility to think about breath and breath biopsy as a way to look for disease which are more distant compared to just the airways themselves. And once every minute, your entire recirculating blood flow goes around your body once. So what we actually do with our device is with each exhalation, we trap and store the chemicals which are coming from the breath. We store them in these tubes here. So by sampling over a minute, you have the ability to look into a much larger volume of blood than would otherwise be the case if you're having to take a blood sample directly. So if you went to the doctor and they said, I've got an amazing test to look for a disease, I just need 4 pints of blood, that wouldn't be the world's best test. Breath is unique. It is the ultimate anytime, any place sample where you can collect virtually unlimited samples. And again, if the goal is to try and find biomarkers of disease at the earliest stage, you need to really drive the sensitivity of the test to pick up these biomarkers early. So there's 2 key parts to the story. The first is how do you get a good sample? And this has been one of the issues that's hampered the field for a long time. So if you think about it, it doesn't matter what analyzer you use to analyze chemicals if you don't get the right sample in the first instance. So what we're able to do with this device is a patient breathes normally, we can watch the breath fractions. So we can say, let's pick out the bit of breath which is enriched from chemicals coming from the blood and trap and store those chemicals onto this sorbent material. And anyone who's been to the doctor and you get a blood sample and it's sent off for analysis, that's called a vacutainer. So we see this as a bit like having a vacutainer for breath. So the ability to collect that sample and then send it off to the lab for analysis. And this hasn't been done before. People have been playing about with bags, blown into a bag, but it's hard to figure out issues like shipping a bag and stuff like that. Things which aren't sexy from a technology standpoint, but are incredibly important if you think about how do you actually bring a test which is useful to the market. And then the second thing is how do I detect chemicals? So this is a cut through of our ion mobility microchips, or a chemical sensor in a chip. And basically what we're able to do is take a mix of chemicals coming in, ionize it, and we apply electric fields in such a way that you tune it to allow the compound of interest to pass through. So you're able to change what you detect just by changing the electric fields, and you can change that just in the software itself. So what you're able to do is just reprogram to say, today I want to look for explosives. tomorrow I want to look for toxic gases, and the day after that I want to look for biomarkers of disease. So it's really leveraging this platform capability of the technology. But what we had to do was make sure that we had worked out the kinks through some of the other applications on the military side of things before you start to use this platform in looking at high-volume clinical trials. So I think if I look, what is it that we're trying to do? Yes, we're trying to develop certain tests, but actually I think the broader vision that we have is breath and breath biopsy as being a new diagnostic category. So in the same way that you go to the doctor to get your blood pressure taken or get a blood sample, for instance, you can have a breath sample collected and you can do targeted analysis of these different VOCs in that. They can give you insight about disease state or what medications that you'll respond to in a way that you can't currently do today. And the key piece for us is that we're having to try and put together different aspects of the value chain. So this isn't just about, my sensor is amazing, it's better than your sensor. It's about how do we get the best sample? How do you make sure the sample is stable to transport? How do you ensure all your QC and quality checks during the analysis are in place? And how do you have the best data science team that can figure out how to get insight from these massive data sets in a way where you can actually develop tests which are useful for patients? And there's kind of three parts to the story. If we look at the hardware itself, so I've touched on sample collection analysis, and I think one of the things that we tried to do is say, okay, this is an interesting and novel approach to diagnosis, but what's it useful for? Where can it be used? And what we tried to do here was work with partners, so people who had a clinical need, who were running clinical trials, recruit patients, collect samples, analyze those samples, and publish those findings. And in doing that, there's been a bunch of papers published all up on our website, which to me gives this tantalizing hint at what you can do using this type of approach. So here it's showing utility of these volatile organic compounds and everything from cancer to infectious disease and inflammatory disease. And the breath biopsy technologies that you need, the breath sample collection and the analysis piece coming together. So to me, this is very encouraging because it gives you that kind of sense of what's possible. But you still need to go from that initial pilot stage through to large-scale clinical trials and validation of those tests. So the second piece is, how do you actually run samples? And one of the issues in the past has been often the kit that you use to analyze breath chemicals would be a £500,000 mass spectrometer that you kind of wheel up to the patient. It's like, I'm going to take a blood sample, but blow into this machine. I promise it won't electrocute you. So the issue there is that while that's good for getting some initial test data, it's not scalable. You can't collect a sample, send it off for analysis. So for the first time, we've had to develop from the ground up, literally, a breath biopsy lab where you can collect these samples and send it back and do analysis of all the chemicals on the breath and then try and correlate that to different diseases. And part of what we're trying to do here is enable the research community as well. So we're trying to say, people come to us and say, I'm interested in trying to pick up Pseudomonas infection in patients with cystic fibrosis sooner so we can get them on the treatment sooner. Is there a VOC biomarker for that? And what we say is, well, we don't know, but we can look. For the first time, we can look in a very cost-effective way where we can collect those samples and go on the hunt for those biomarkers. So it's really trying to enable these clinical and academic researchers who understand that clinical need and trying to give them the tools and technologies to see whether or not there's benefit that can be brought to patients. So we're trying to piece this together into the third part, which is about development of tests. So you have your foundational elements of the technology, the infrastructure in the labs and capability that you bring together. But ultimately, sitting at the top of the value chain is the test that you develop and deliver for patients. So we focus, as I said, on two key aspects, which I'll touch on in turn. The first is early detection, which is picking up cancer early, simply. And then the second is precision medicine, which is getting the right treatment to the right patient at the right time. So I want to start looking at detection of cancer and the kind of why. And I've used the slides dozens of times, and it still shocks and surprises me, this stat. 1 in 2 of us are going to get cancer. Killed 14 million people last year. parents, partners, children, grandchildren, 1 in 2 are going to get cancer. And when you look at the kind of battlefield, it's very uneven. Survival chances are very uneven and different. So, on the one hand, some cancers are highly treatable. Your 5-year survival rate is very good. So screening works, treatments are effective. Other types of cancer, like pancreatic cancer, are extremely deadly. There's been very little progress made there. But the relative incidence is, not as large as some other cancers as well. So, where we see the biggest immediate need is around lung and colorectal cancer, where you can see in lung cancer over the last 40 years or so, there has been barely any progress in 5 and 10-year survivals. So a lot of the therapies which are being developed, the kind of advanced biological therapies, are extremely expensive, but they don't cure in the long term. They will extend life by a few months, but they're not a cure. And the crazy thing is, we already know what to do. Detect it early. So there's no better example than colorectal cancer, where if picked up at stage 1 is about 95% survival chance, and if picked up at stage 4, it's about a 5% survival chance. So if there's a single lever you could pull to change things, it would be detect early. So I want to just talk about a couple of examples of the work that we're doing within this. The first is our Lucid lung cancer trial, which is looking at these volatile biomarkers in patients with lung cancer. So, in the UK, lung cancer costs £2.5 billion, but kills, 43,000 people are diagnosed each year, and the vast majority of those, unfortunately, do not survive. Now, we know early detection will work, but today, only 14.5% of patients are detected at that early treatable, survivable stage. And a lot of that is by incidental findings. So when we're out speaking to clinicians, I was like, what's an incidental finding? That's, you're in a car crash, you break a rib, they scan, and they're like, what's that? So that's not really a long-term sustainable strategy for screening. Break your ribs when you get to a certain age, and that's how you're going to find it. So what we know is that if you could increase from 14.5% to 25%, you would save close to 10,000 lives over a 3-year period. And that's an arithmetic fact. So what we now need to do is have the technologies that can help shift the dial on that rate of early detection as relatively modest increases, 14.5% to 25%. So we're running a trial at the moment called Lucid, which is funded by the NHS. And what was wonderful, the support that we got there and it was through SBRI scheme. So we kind of, had our core business and technology focused on military. And when we were looking at these types of applications, it's, how do we basically incubate and build the medical business under the umbrella of the main company? And what was really important for us was winning contracts like SBRI funded by NHS, which give us the kind of time, space, funding to try and explore these types of applications. So, this trial has gone well. It's now recruiting over 26 clinical sites all across the UK. So up in Papworth Clinic, we have a lot of patients coming through clinic there, recruiting up to about 3,000 patients. And the goal here, again, is what are these biomarkers and how good are they at picking up early stage disease? And when we start to think about, well, how can I fit this into the clinical pathway? Because I think for us, we were technologists coming at it from the point of view of, we know how to detect chemicals, we think we understand the need there, but trying to understand how it can be deployed within the clinical pathway, we didn't have a lot of experience there. So a lot of the learning that we've had is trying to figure out how you can bring these technologies to the right point within the clinical pathway so it can be beneficial. And there's kind of 2 main areas in lung cancer that we see being a big target today. So I mentioned in the UK, we don't have screening for lung cancer at the moment. It's all done by incidental findings. In the United States, they do have screening. They use something called low-dose CT, which is basically, take an image of your chest and try and find a suspicious lesion within there. And this approach, it showed that you could reduce relative mortality by about 20% of lung cancer, but you're dosed with radiation during the test. It's quite an expensive test, about $285 per test. But the thing that is worst of all is the false positive rate. So over the lifetime of this study, about 53,000 patients were part of the study. About 40% of patients had a positive finding. And what's the number? 96.4. It was a false positive. So as you come through the door, there's a pretty good chance you're going to have a positive. And if you're positive, it's a really small chance that you've actually got disease. So, one of the big factors that we need to think about with all of these types of early detections and screening technologies is not just, can you pick up the disease? It's what's the false positive rate look like? What's the selectivity of the test? Because the vast majority of people coming through the door for test don't have cancer. So we need to have technologies which are both sensitive and selective. So what we're trying to do here again is think about, how can you fit into, existing clinical pathways? So again, just to look at some of the numbers that underpin this. So 1 million people who get tested, over 250,000 will have a positive result. And then when you say, well, what happens then? Quite a high fraction of them go for repeat imaging, about 90%, which is, expensive. Patients are really worried through this process. But a really significant fraction go for invasive procedures. So it can be biopsy with the harms associated with that. And an unbelievable proportion go for surgery as well. So you get a resection, what turns out to be benign disease. So I think here, the goal is to try and think about how can new technologies, not just our technology, but a bunch of the other molecular diagnostics in development today, how can we augment what we already have in place to deliver better outcomes for patients? And reduce healthcare costs overall. So the goal here is where you think if you have a positive finding, rather than have the biopsy, rather than have the surgery, see if there's other breath-based biomarkers that will confirm or not whether or not there's likely to be disease present. But where we see the biggest potential, again, comes down to this early detection. So when you think about the type of test that hypothetically you could deploy in a GP surgery, in a pharmacy, it needs to be non-invasive. I hate needles. I work in this industry and I still go to the doctors and it's like, I want to take a blood sample. I'm like, see you later. So we can't underestimate that aspect of patient acceptability and compliance to the ultimate performance and acceptability of these tests. They need to be relatively low cost as well. NHS does not have a lot of money. Other healthcare providers do not have a lot of money. And if you're thinking about large population-based screening, the test itself needs to be low cost, and also it needs to be able to deliver on the sensitivity and specificity promise. But that being said, tests don't need to be perfect. Getting a positive finding here isn't the same as saying you definitely have cancer or don't have cancer. You're part of an overall diagnostic chain where you can be referred for follow-up imaging as well. So the next piece I just wanted to talk a little bit about is on colorectal cancer. So colorectal cancer kills 215,000 people annually. It killed my wife 2 years ago. And here we know, again, the early detection is the key, 95% survival chance. And despite the fact that we do have screening here, not enough people are detected at early treatable stage. And when you look at what's the problem, again, with my engineering technologist hat on, I was thinking about how good's the test. But actually here, the nature of the test also matters massively as well. So at the moment, colorectal cancer screening is either a fecal-based test or a colonoscopy. Who here would rather have one of those two compared to a breath test? So, when you look across Europe, compliance rates are low. People do not like doing the test. And if they don't like doing the test, and you try to unpick what this means, so 100 patients who have cancer, half will not show up for the test. Of that half, because the test itself isn't very sensitive, it's not that good at picking up cancer, a large fraction of those patients, again, will go on to develop late-stage disease. So there's two problems here. The test has to be acceptable, and the test has to be sensitive without sacrificing selectivity. So one of the early pieces of work that was done by one of our clinical partners up at Coventry Hospital, which is looking at VOC biomarkers. This was in urine, actually, so you get these VOC biomarkers in breath and in urine. blood being the key linking compound there. What they're able to show was two things. One, patients don't mind the test. Two, the sensitivity was very good as well. And again, it's when you compound these factors together. So those 100 patients that have disease are now happy to show up for the test and are now able to pick up disease because that sensitivity is a lot better. So this was one of the early results that was key for us to think, and we want to have a go at trying to make this technology work in diagnostic applications. So again, what that translates to is early detection saves lives. And when you look at what the impact is, and there's kind of two ways I look at this. And the way I see what we're trying to do in the company is, if we have a mission to save lives, a successful business will be built off the back of that, because ultimately that's what healthcare payers are going to pay for. And when you start to think about who is it that should be screened, it's largely driven by age and risk. So in colorectal cancer, anyone over the age of 55 basically should be screened. So in the US, that's close to 90 million people. Globally, it's about 950 million people who should be screened for colorectal cancer, and a single-digit percentages who are today. So again, to me, what this highlights is we know the solution: detect early. But this whole set of problems you need to solve in terms of compliance, acceptability, test performance, economics of the test need to fit together. I'll skip through this quite quickly. So this is more we kind of show the investors like why we're better than everyone else type thing. But again, just to summarize it in one slide, again, it comes down to the question I'd ask you. Would you rather have a breath test, which is completely non-invasive, and then go for follow-up tests off of the back of that? Now, one of the things that's interesting in cancer detection at the moment is actually the amount of investment that's going in these days. One of the aspects that we've learned is that early detection is as much of a financial engineering problem as it is a science and technology engineering problem. And the reason for that is if you think about the length of time it takes to run clinical trials and the cost to run those clinical trials, it's a difficult business proposition to frame together. You have these kind of big markets out there, but you need to go to investors and say, some of them, the likes of Grail, I need $1 billion to run clinical trials. And they're going to be running a 120,000 patient study. So I think it is a challenging business to be in, but what is exciting from my perspective is there is investment going into the field now. The problem is acknowledged. I hope we're part of the solution, but the most important thing is for someone to solve the problem. So a lot of these companies here are looking at liquid biopsy type approaches. So this is looking at fragments of circulating tumor DNA that come from a tumor and then are picked up in the blood. And if you're thinking about treatment selection in cancer, it makes a lot of sense. Often, later stage disease, you have a particular target mutation that you want to hit with a certain drug. It makes a lot of sense. If you're thinking about early detection, it's quite challenging because often you don't get detectable levels of circulating tumor DNA in the blood at that early stage. And that's not a question of better technologies, that's more of a question of tumor biology. You just don't get those detectable levels. So I think the hope that we have is that we can be not necessarily instead of, but in addition to a lot of the approaches that are also being developed alongside what we're working on. And we're very excited to announce just about 2 months ago that we've extended the scope of the trials from the initial lung cancer focus into our Pancancer trial, which is looking at multiple tumor types. Again, the goal is trying to find these VOC biomarkers associated with different cancers, and we'll run this in conjunction with cancer research here at Cambridge University. So a lot of the kit is going down to Addenbrooke's, and then the goal there is to try and find these biomarkers across a broader range of disease. So I just want to finish the last couple of slides thinking about, I talked about the first big problem in healthcare. We detect disease too late, which is particularly acute in cancer. The second big problem is too many people are on drugs that don't work for them. So globally, we spend $1 trillion a year on drugs, and 40% of patients, it doesn't bring benefit for them. So the overall goal and promise of precision medicine is trying to match the right patient to the right treatment at the right time. And here, breath VOCs have an important role to play. What we're trying to do is find, the best match between the capability and then the disease of interest where you're trying to match some of these target therapies. So we tend to focus more on inflammatory type disease on the respiratory side. That's things like asthma. And then on the cancer side of things, this is less to do with what's the right therapy and a little bit more to do with some of the side effects that you will get with some of the emergent therapies. So things like immuno-oncology, which are exciting drugs. Again, I don't think they're cures for the long term, a lot of them. And a lot of these therapies come with significant side effects, life-threatening side effects. So as well as trying to think about what drugs work for me, what are effective, I need to think, what drugs can I take without risking myself further? So, I just want to pick on one example here, which is therapy selection in asthma. And asthma was interesting for me. Me and my close family don't have asthma. I didn't know anything about it. It's obviously incredibly common. 5.4 million sufferers a year. But the thing that surprised me is the treatment is completely trial and error. So at the moment, say you have asthma, I'm going to start you on the cheap drug. If that doesn't work, I will up the dose. And if that doesn't work, I will move you to one of the more expensive drugs. And up along this escalating pathway here, when you get to these step 4 or 5 in what's called the GINA guidelines, you're using treatments which are extremely expensive here. So some of them, Xolair is a $1.8 billion a year drug for Novartis, and 30% of patients don't respond to it. So again, if you think about it from the point of view of the NHS and the payers, I don't want to have to pay for drugs where 30% of patients aren't going to respond to it. I need to have better tools and technologies that will say, this drug will work for you. And it's a problem that's going to get worse. So even just in asthma, as we have whole new sets of therapies coming along which are being developed, and again, it's trying to understand what the underlying inflammatory subtype of these patients are so I can get them onto the right treatment first time. And the goal here is to use the same paradigm. So you want to use the same breath collection device. So it's the same device that you'll be using to collect a breath sample if you're looking for lung cancer biomarkers, or if you're down in the severe asthma clinic, you're relying on this device, again, to collect the chemical markers. And we're looking for different VOCs, which are giving you different information instead of diagnosis that's saying this therapy is going to work for you. So the idea, again, here is thinking about breath as a new diagnostic modality and building up the infrastructure from the sample collection through to the analysis and the reporting there. In a way that makes it much more broadly applicable and accessible to as many patients as possible. And I think there's an interesting byproduct from all this work that we're doing, both in trials that we run, but also what we do for our partners. So clinical researchers, academics, or pharmaceutical partners, where actually the true value in a lot of these things is derived from the data that gets generated. So today, through the studies that we've run, we've already built the world's largest digital biobank of breath biopsy profiles where we have these patient populations. We understand a lot about them, not just whether or not they have lung cancer, but do they have COPD, what medications they're on, how they respond to those medications. And we analyze absolutely everything that's on breath. So the lab that we have over on the other side of Cambridge has, GCMESCO spectrometers from floor to ceiling. Not quite, but kind of wall to wall, which is all around trying to get as much insight from these samples as possible. So again, we provide the foundation for the next generation of tests, in addition to the ones that we're focused on today. And again, it's really trying to deliver this vision of breath as a new diagnostic modality. And Owlstone, little Cambridge company, we want to be the global player in this. So we have global ambitions for what we're trying to do here, and Cambridge is a key part of our historical story and where we want to continue to focus. And again, what underpins it for me is the impact that early detection can have. And, it's not just me that's lost someone. Almost everyone in the company, almost everyone in this room will have a similar story. Again, I think the key piece is that we have to try. Us, others that are involved in, trying to develop better early detection technologies. Some will work, some won't work, but we have to try because we know what's at stake and we know what the output would be if we can detect at that early stage. So that's a little bit about us. I would love to answer any questions that you might have about any aspect of this or more generally about the business or the history of the company.
Well, Billy, thank you very much. I can hardly imagine a better example of the application of research for something really worthwhile. So now Q&A. So that Billy can concentrate on answering the questions, I'll just choose the people I like from the audience. But maybe if you put your hand up, I'll try and call you in, and with any luck, our splendid microphone handlers will get to you before your turn comes. But let's start with the gentleman over there.
Thanks very much. That was fascinating. Your test unit, can it do effectively multispectral tests, or does it have to be tuned for the specific VOCs for each test in turn?
Yeah, no, it's a good question. So we deliberately separated out the sample collection piece. So the sampler will collect all the chemicals on your breath and pre-concentrate them over time. And then when you send them to the lab for analysis, you can do one of two things. So you can either run that targeted analysis. So I know what I'm looking for, these 5 chemicals, and you program it to look for that. Or you can do a more untargeted analysis where you say, right, I want to look at everything that's present, and then match that to a cancer or a healthy, for instance, to try and find, well, what are the features or biomarkers within the chemical signature that say cancer, not cancer. And we use a mix of lots of different analytical approaches. There are, mass spec, FAMES, you name it. What you're trying to do is get that as much understanding about, the mechanistic basis, but also the chemicals that are there. And then once you have that, you can functionalize into that targeted assay, which is what allows you to crank through a large amount of patient samples.
Your chip, is that basically a mass spectrometer?
Kind of. The principle is somewhat similar. So it separates ions according to their mobility, which is basically how fast they move in an electric field, which depends on mass of charge, but also things like cross-section. So it's a bit like a mass spec on a chip.
Right. So what are you actually measuring then? Is it molecular mass? Are you looking for a particular?
So it's the differential mobility, which is basically how fast something moves through an electric field at high field compared to low field. And that in turn is determined by the mass, but also things like the cross-section of the ion as well.
Right.
Thank you. Yeah, it's a really interesting talk here. I wondered, how do you decide what you're looking for to diagnose different diseases? Is it purely empirical, or do you have some hypotheses that you can then go out and test?
That's a very good question. I think it's a mix of both. So I think for some, like if you look into the literature, people have been trying breath stuff for quite a long time, actually, about 30 or 40 years. And like all papers say the same thing. Breath dates back to the Greeks, and the modern breath era was back with Linus Pauling blowing into a mass spec in 1971. And, I'm like chief skeptic in our business. So when we were doing all the security stuff and we're thinking, well, should we look at medical applications? I'd be like, well, there's tons of papers out there. None of them say the same thing in terms of biomarkers. And if it's so great, why is it not being used in clinic today? And I think there were some foundational elements that needed to be addressed. Basic stuff like how do you get a good sample? And then, what are the right mix of analytical tools that you can use for, discovering biomarkers? And it's only when you have those elements in place that then you can start to go out and think, well, my untargeted analysis, which is more empirical, saying I analyze everything and I try to find features that relate to lung cancer or healthy. So only then you can do that type of stuff. But then on the other side, where you do have more of a mechanistic understanding, then you can narrow the search down and say, right, I'm going to look for these compounds to see whether or not there is a signal that explains, the difference between those categories. And one of the things that's relatively new and to me really different about this was that when I showed that slide thinking about where do these chemicals come from, so a lot of them are metabolites that originate in your body, but actually you have tons of chemical noise as well, which is I breathe in chemicals when I'm cycling across Cambridge, and I breathe chemicals out again. So those are exogenous compounds, and so is food stuff. I eat a biscuit, and you will see byproducts of that. And we used to treat that like chemical noise. So you need to have the analytical performance where I can see the signal of interest, ignore all the other crap that's in the background. But some of the recent work that we've seen that's been really interesting was around a compound called limonene, which is everywhere. It's in fruit. You eat it, it's in fruit. So it's not an endogenous compound in the body. It's not produced in the body. It comes in externally. But actually, patients that have early-stage liver disease can't metabolize it properly. So you start to see elevated levels of it, and because it's lipophilic, it goes into the fat, and then it absorbs out of the fat as well, even post-liver transplant. And for me, that was a real eye-opener, because beforehand, we had been doing analysis thinking, oh, it's not an endogenous metabolite, get rid of it, it's junk. But actually, by exploiting the fact that something's gone a bit wonky with that part of the body, you actually have a marker which can be useful in diagnosis, which is this, what would otherwise be classed as, chemical noise exogenous compound. So I think it's an exciting time because once you have that kind of foundational platform to start to believe the answers that you get, you can start to, get your hands around these really interesting research questions to say, what are the markers in this broad class of different disease? It's a long answer.
Hi, Billy. Not the question I was going to ask you originally. It's a different question. If you're going to do breath analysis, you really have to have a baseline for that person.
Yeah.
Is there any group, CEN, ISO, anybody who's trying to write a standard for breath analysis?
So I agree that one of the most useful things about breath, one of the things that you need to do is think about yourself as your own baseline because you have your own metabolome. So actually looking for differences and deviations from your own metabolome is interesting in its own right. So we've tried to do work around the standardization of the sample collection piece because that's your entry point into the field, if you like. But I'm not familiar with any group that's doing longitudinal sampling.
I'm in CEN 264, working group 41, and we're actually working on odors, et cetera, and odor profiling, so we got to have a chat. The other one is, I thought you were FAMES. Since when did you become a GCMESCO?
So again, it's interesting when you think about... Yeah.
Could you tell us what those words mean? Because we don't
Yeah, so FAMES is the microchip. So again, it goes back to the question that was made earlier, the difference between untargeted discovery versus a targeted assay. I think when you look at biomarkers generally, breath specifically as well, you have different analytical tools which are better for different jobs. And you need to think about solving each of the problems within it. And I think what the breath field has done before I've developed a sensor, it's amazing, my sensor's better than yours. But then they haven't thought about how the hell do I get a sample to my sensor. So I think the point I was trying to make with the overall value chain was thinking about breath biopsy as a diagnostic category that has multiple technologies within it. The sensor is one part of it, or the analytical piece is one part of it, but that's not the whole of it. You need to solve all of the issues collectively, otherwise you get nowhere. You get into this domain of doing these small pilot-based studies, which are utterly, you can't reproduce them at all.
Do you have any good news for people who have got brain diseases?
That's one of the categories that we're looking at in the Pancancer study, so for brain cancer. And what we find that's interesting on the services side of things is where, again, once you kind of pose the question of other VOC biomarkers for different types of disease, brain disease, for instance, you don't necessarily know a priori that there is. But I think what we can do now is go try and find them in a way which is cost effective for the research community. So we do have some programs which are being put together, not by us, but by external partners who come in and use the platform to try and answer those types of questions.
Hi. The examples that you've given are to do with diagnosis of physical diseases. Do you think there's any possible application in terms of identifying potential mental health issues? And also, do you think there are possible applications in terms of animal welfare for use by vets?
Yeah. So the first question, the short answer is I don't know. And again, it comes down to, is there a detectable VOC signature? Is there some type of mechanistic pathway that leads from a particular disease or disorder that is reflected in some type of chemical change or metabolism change? So again, the answer to that is I don't know, but we can enable researchers to find out if there is. But our focus in terms of how we can apply our internal resources tended to be around early detection of cancer. And the same kind of goes on the animal welfare side of things. Again, animals have a volatile metabolome as well that gets disrupted with disease. And through the research tools and services side of the business as well, we've had interest in everything from cows to chickens. So, which again, it's trying to solve specific problems. But once you give someone a new tool to play with, they're like, right, what nail can I hit today with this? So I think that's what we're trying to enable.
Right. Well, thank you very much. Superb. There are two ways of protecting intellectual property. One is by patents, and the other is by running faster than everybody else, so you stay ahead. I wonder where you feel the balance is with the work that you've been doing.
I think it's a bit of both, actually. So I think you're right. Where you have bits of hardware like your entry point into the game, if you like. And we obviously, with all investment that we raised, the IP and how we protect is important part of that. But actually, again, one of the things that you see with the dynamic of creating data through running trials, so you have a bit of a virtuous circle there. So if you have more data initially, you have slightly better insight about where's the regions of interest of the chemicals that relate to a particular disease, which means you can commercialize those faster. So I think certainly for us, we have today the world's largest digital biobank of breath samples. And I think that with the lead that we have, we've no intention of giving it away anytime soon.
I think you may have answered this question partially before, but if one is applying this system to regular GPs, you want something whereby the individual can go to the GP with no symptoms at all and have a routine check, and therefore there must be 2 or 3 compounds that are common to all the cancers that you can determine? Is that possible?
So that's part of the rationale for the Pancancer trial, actually, because if you think about the mechanistic basis of cancer, so you're saying it has an altered metabolism. Let's try and find the altered metabolic markers, some of which are likely to be common between different cancer types. And then when you look about some of the site-specific markers, for instance, on colorectal cancer, you have the microbiome being disrupted as the tumor grows. So you have site-specific markers which form a contribution, which is a tighter fingerprint, if you like, for that particular cancer type. So I think we'll be able to, again, uncover some of those questions through running these larger scale trials across different cancer types. But I completely agree with you that the goal is not to do the test when someone has, an alarm symptom, but it's when they're in a risk category where you say it makes sense now to do a screening type test. So in the lung cancer example, for instance, the screening criteria is anyone over the age of 55 with a 30-year smoking history. But actually about 50% of lung cancers are outside that predefined screening criteria today, which relates back to the test performance, the test economics of what they have today. So the goal is to have something which people are happy to do in a GP type setting, but it has sufficient sensitivity and specificity that you pick up the early stage cancer so it's treatable without flooding the healthcare system with a gazillion false alarms.
I was intrigued to see in your list of various diseases, celiac disease, having a personal interest in that. How easy is it to detect chemicals in the breath of patients with celiac disease? And if they then go on to a gluten-free diet and therefore controlled, do you then still detect that they have had celiac disease? Because there is a blood test, as you're no doubt aware, but it's not necessarily 100% positive, and you're faced with a jejunal biopsy to really determine that.
Yeah, no, that's so in that slide, that was one of the applications that our clinical partners are looking at. Again, the approach of trying to say, here's a tool, see what's possible with it. So that was one of the programs of work in clinical studies that was done by Coventry as well in that case. So they've got it up to pilot stage, and then what we try to do is say, okay, is there real clinical need here? Is our test going to make a difference? So you're looking at, are there other tests? How good are those tests? What do the economics of that look like as well? So the kind of triaging process that we went through afterwards said early detection is where we're going to be focused on as a business. And then this wider set of opportunities we'll pursue through the partner network and academic network as well.
Billy, so if I kind of think about the problem that you're tackling, the detection problem, intuitively it seems to me to break down into two parts. The first is, can you, for example, from patients with severe disease and using the most expensive analytical technology, detect and identify potential biomarkers and establish what their chemical composition is and so on, or what's the mixture of those. And then the second part, intuitively, is can you then make that available at lower costs in high volume for early detection? So the question really is, has that first part been solved or essentially solved by looking at those that are severely ill and using other detection technologies? Great presentation, by the way. Thank you very much.
So that's what the large-scale clinical trials are designed to answer, that exact point. So Lucid is set up to answer that question. Because I think what we've seen in the literature before, again, all these papers have been published, especially around something like lung cancer, is it seems like there's a signal there because people have been publishing, but there's little agreement between them. So I think it's a case of, it seems there's smoke but no fire quite yet. And the only way that you can definitively get the answer to that is running these larger scale studies, which I think, again, on the Lucid example, is the world's largest study of its kind ever undertaken, because that's the only way you get that type of definitive evidence.
So, for example, my assumption that those that are severely ill will present a higher concentration of biomarkers, that's actually an incorrect assumption. Is that what you're implying?
So I think it definitely makes sense to a degree. So the only difference being that with later stage disease, you have more systemic change as well. So things like weight loss and those types of things. So, it would be slightly dangerous to extrapolate from late stage back to early stage. It's just a specific marker panel. So I think you do need to have earlier stage disease as part of the cohort that you're looking at initially, because it's that translation from late to early, I think, would be a bit of a risky jump. And what we do in the Lucid study is, because this patient's coming through the normal clinical pathway anyway, you have, so it tends to be patients who do have some type of symptom or incidental finding. So it's a slightly enriched population. That means you have more cancer-positive patients in that population, which means the trial can be run for a shorter time period to accumulate enough patients. But actually, because of that, there's also a higher fraction of patients with early stage disease within that same overall trial population as well. And this was one of the interesting things about our transition from bunch of chip guys flogging stuff for military applications to trying to think about, well, how do you go through this entire analytical workflow, but then think about the additional skills that you need to bring into the business, design of a clinical trial, running of a clinical trial, all the aspects that we didn't have that kind of native expertise. And that's why fundamentally we decided we need to split this out into a separate entity. You need to have a clinical operations team. You need to have all the things in place that we didn't have in place prior to that.
Yes, thank you very much for a very, very interesting talk. My question is your method, might it be suitable for analysing hormonal levels and imbalances in people, otherwise healthy people, specifically estrogen, and that's both for analysing hormonal imbalances as itself a precursor to other kind of pre-markers, and second for the transgender community for whom currently the blood tests are incredibly inefficient?
I don't know is the short answer. So again, the kind of theme from earlier is we are set up to answer those types of questions when they come through, trial through an academic partner or whatever. I think when I think about the main limitation of the approach is that you're looking at chemical markers, but they need to be volatile enough that they are getting generated from the airways coming out or coming from the blood out as well. So that means that it won't work for everything. So that condition of there must be a volatile compound needs to be satisfied for any particular disease category that we're looking at, which won't be everything, but again, it will be a lot of things.
A couple of questions, if I may. First one, what's the turnaround time for patient breathing into the device to having a result? And the second one, what's the regulatory pathway for a device such as this?
Yeah, no, so the first one was interesting, actually, because we had our little, famous microchip. So our original concept was, this is easy. We stick that in there, people breathe into it, and you look for your targeted compound. But actually, the health economics and the regulatory experts that we worked with, it came through the SBRI program, actually. So they came in and they said, you have a few challenges here. If you're thinking about GP-type deployment, for instance, in cancer, you don't need a real-time measurement. So if you collect a sample, have it analyzed, and the answer comes back the next day, that is not going to impact your survival chance. You need to pick up early-stage disease because it's most treatable, but it doesn't need to be a real-time measure. And this was important because it means that you can then think about your analytical piece off somewhere else, which dramatically simplifies the regulatory pathway, but also makes the economics work as well. Because if you're sending a GP, we've got an amazing widget, sniffs chemicals of different disease, it costs 500 quid, 1,000 quid, whatever. They don't have that money. So trying to think about how you have a new modality, but it fits into kind of pre-existing notions of collect a sample, send a sample off to analysis, was the kind of conclusion that we came to working with the health economists and the regulatory experts as well. So it means that, patient will breathe in to this. So it's configurable in discovery, because what you don't know if you're trying to find a biomarker for a particular disease, if you sample for too short of a time period, and if you didn't see anything, it's hard to disentangle the answer of, is there a biomarker, yes or no, or did I not sample for long enough? So what we tend to do in the first instance is sample patient's breath for a few minutes. So you pre-concentrate all the chemicals coming from blood, which is recirculating once a minute around your body, which gives you the best chance of finding the marker. And then once you know the marker, you can run it for a few tens a second to collect a breath sample, sent to our lab for analysis. And that's where, back to David's point, we have a lot of the kit and infrastructure thinking about, higher throughput type analysis, where you can get the answer back the next day, which is completely fine from a clinical standpoint. And again, it means that this becomes a class 1 device and is simpler from a regulatory pathway standpoint, but also the scalability of the approach into different clinics and applications as well. Once you're thinking about this is collection and then separate analysis back in lab, looking at different indications which are developed over time.
Halfway down the aisle, the grey, sir. Left a bit, down a bit further. Gentleman just on your left now.
Does mesothelioma produce a particular signal? Knowing how difficult, how people who have asbestosis, it can take many, many years for it to appear.
Yeah.
How sensitive is it in picking up people who've been exposed to asbestos? Is this a mechanism by which you can get early detection?
It is a subset of the population we're seeing in the Lucid trial, actually, but it's a smaller subset compared to the other cancer patients which are coming through. So I don't think we will get sufficient data in the current trial to say anything definitively on that.
Hi. I had maybe a kind of wacky thought. So if FAMES is partly sensitive to molecular mass, would it be worth playing with isotopes? So if you give someone some water or glucose enriched in oxygen-18, will you then get some metabolites half an hour later with a peak at a slightly different place in your FAMES spectrum because of the different molecular mass?
Yeah, so I think there is a breath test that has been developed along those lines. I think it's H. pylori or something. Do you know? Yeah. I think there's one breath test which is along those lines.
Okay, so that's stomach ulcer one.
Yeah,
okay.
Yeah. And I think what we're now a little bit more switched on to, back to the limonene example,
Yeah, I was thinking that as well. Yeah.
was that exact thing of, if I understand mechanism, what can I take, which would then result in something which is only expressed if I have disease because the pathway's been altered in some knowable fashion. Right. So I think that's a really interesting area for exploration. We're working with the Cancer Metabolism Group here in Cambridge as well, to try and go from that foundational understanding of mechanism through to something where you think, there should be something popping out here.
Right. Okay, thanks.
Well, ladies and gentlemen, right, last one.
Well, thanks very much. A riveting presentation. I'm interested in the doggy bit, because really most of us want to know whether we've got cancer or not. And if the dog is as precise as it seems to be, can that be synthesized in some way by, first of all, I assume it's a molecule that is coming from the breath that the dog is sensing. It's 1,000 times more sensitive than our nose or something, I believe. So could something like stem cell research with, in other words, biomechanical approach to this possibly produce something which tells us, because diagnostically brilliant what you've got to actually diagnose what we've got, but just to tell us initially yay or nay in quite a quick fashion, and whether there is research in this direction that you're aware of, or maybe someone, maybe a brilliant vet that's locally is already investigating this.
Yeah, no,
anyhow, many thanks.
There's a company called Medical Detection Dogs, which is aimed to try and use dogs to pick up cancer. And I think to me, it fits into this interesting patchwork of evidence suggestive that there are markers, related to cancer and other diseases, which are these volatile organic compounds. So for me, it's part of the evidence base, if you like, that we should make investments to try and find out what these markers are, how you can turn that into a scalable test. And I think that's part of the challenge with the dogs, for instance, is there may be some evidence there, but the dog can't tell you that it's butadiene or pentane or whatever it may be. So it knows what the smell of it may be, but it can't tell you what it actually is. So I think you do need to understand the actual target compounds and how well they perform in these larger scale blinded studies. And then you have a second kind of practical issue of how do I deliver a test to 950 million people who should be tested? And I think what we're trying to do is capture what the dog has on a silicon chip, so you can deliver a test which is acceptable to patients, which is this easy screen triage type test for our early cancer detection that doesn't poop on the ground.
Well, thank you for bringing us back down to earth, Emily. I don't like to stop the proceedings, but we've kept Billy on his feet for a long time. It's customary at this stage for somebody to stand up and drone on for hours and hours about the structure and the elegance and the flow and the beautifully illustrated talk and the fact that it all really matters. But I think you've heard that. So please can we thank Billy in the normal way.